Hello, everyone, and thank you for the opportunity to talk to you today about non-compartmental and compartmental approaches to pharmacokinetic data analysis. My name is Paolo Vicini, and I would like to acknowledge also my colleague, David Foster, who originally was responsible for this content when the course was put together at the beginning. Here's my current disclosures. And I think it's helpful to start with some background in terms of the words and the glossary that we're going to use in the remainder of this presentation. So we refer often, of course, to pharmacokinetics, which in a very simple way is what the body does to the drug. And then to place this in the proper context, we'll also sometimes talk about pharmacodynamics, which is what the drug does to the body. And this is all in the context of temporal disease progression and measurable therapeutic effect that a drug has a goal to modify. And running alongside this on the background is the idea of variation. So the data that we measure are subject to noise and variability, and so they're not necessarily a perfect picture of the biological system that we are trying to estimate and to study. So PK and PD. PK, again, is what the body does to the drug. It's fairly well known, and it's required to understand pharmacodynamics, PD, which is actually part of the ultimate goal of drug discovery and development, to understand what the drug does to the body, which is sometimes unknown, often unknown, but has true clinical relevance. And so the questions in this area that we're going to tackle relate to both PK and PD. And like I mentioned earlier, PK and PD are based, they operate or they are based in the context of disease progression. So a certain time course of the patient's status worsening that the drug would like to impact and to change. This is the broader context of what we're going to talk about. But in the remainder of this presentation, we will talk about pharmacokinetics, so PK. So PK can be very complex. A drug in the body is constantly undergoing change. Concentrations in various body spaces rise and fall depending on the particular characteristics of the molecule and the biological system that is being studied. Because this behavior is so complex, there is a need to summarize it in a way that can be easily communicated and that can be used, for example, to compare molecules one to the other, or to properly evaluate and estimate the effect of a certain drug or a certain therapeutic. And so that's how these complex curves, this complex behavior can be summarized in terms of parameters that can be derived from these time courses. So a PK parameter is something that we can use to summarize what is a very complex behavior, time-dependent data, that is due to the interplay or rates of distribution, elimination, accumulation. These parameters are informative on the characteristics of the drug molecule, and they can be descriptive or observational, or they can be quantitative. So we'll actually see what the difference is between these in a few minutes. And to estimate these parameters, we use mathematical models. And the two important classes of these models are the compartmental modeling class and the non-compartmental modeling class. And so as far as the goals of this lecture, what the goals of this hour or so that we spend together are, is that we'd like to understand a little bit what these model assumptions are, what are the assumptions that inform every one of these models, what are the parameters of interest that we distill from PK curves, and what to expect from these estimates. And so we, by the end of this time, we should be able to understand clearly what each model assumptions are and how we can choose the most appropriate method depending what information is required and what data are available. We're not going to make any conclusions in terms of which method is better than one another, because of course the answer to that depends on the question being asked and the context of where the model is being used. So we talked about PK and what it means for a drug in the body to be constantly undergoing change. A drug molecule, whenever it's administered, follows processes of absorption, transport in the circulation or across membranes, transformation via biochemistry, and elimination. And you may have heard the moniker ADME, absorption, distribution, metabolism, and excretion, being used to refer to this collection of changes. And like I mentioned earlier, depending on the space where the drug is introduced or where it gets and how it's transformed, drug molecules describe or undergo several changes. And so the concentration time profile, depending on how the drug is given intravenously, intramuscularly, orally, and where it goes, can have several different profiles, several different shapes, some of which can be very complex. And so we refer to this collection of time and space dependent changes as kinetics. So kinetics in general, not just for drugs, but in general for any substance in a biological system, refers to its temporal and spatial distribution. And specifically for drugs or for xenobiotics, pharmacokinetics is the collection of temporal and spatial distributions of a drug or drug combinations in a biological system. So some of you may be experts in pharmacokinetics. The definition of kinetics to cure is a bit broader because it doesn't just refer to changes over time, it includes also changes in space. And so a complete description of these concentrations that we have described requires both an understanding of spatial distribution and temporal distribution. So where in the system and when in the system. Now, you can appreciate how if we were going to describe this formally, writing the equations of this behavior in spatial and temporal detail is quite demanding. In fact, it's quite difficult. For the mathematically inclined among you, it would boil down to write partial differential equations, i.e. rates of change, for the drug molecule in space and time. And however rigorous, it's not really practical. So while people over time have tried to develop spatially realistic models of drug distribution, they soon realized that this requires very detailed knowledge of things like physical chemistry, thermodynamics, and circulatory dynamics. And even if you are able to formulate them, they can be very difficult to solve. And to top it all, it's challenging to design an experiment that can help us quantify the parameter values for these time and space behaviors. While desirable, often not practical. So over the years, the discipline has developed several other approaches that are a bit more tractable to describe change over time and space of drugs in biological systems. And one that is now the state of the art is what we call physiologically based pharmacokinetic models, or PBPK. PBPK models are a compromise between spatial realism and temporal realism and tractability, and essentially they boil down to creating differential equations for the various organ systems that a human or animal body has, and then building the arrangement between these organ systems in terms of circulatory flows, so that one can write organ-level realistic kinetics. And this is not a fully spatially distributed model, but it is a good compromise because it boils down the system to its most important components, which are the organs where the drug distributes. And like I said, this approach is now state of the art, and one can also use it, for example, to translate among species, say from preclinical studies to small human studies to larger human studies. And because they are mechanistically more realistic than other models that we see, they lend themselves well to translation and to translation between species and other settings. There's also, as we go from more realistic spatial distributions to something that is a hybrid, which has organ-level kinetics, there are also ways to sort of lump the components of the system empirically based on their behavior. And when I say behavior, it can mean time, location, or a combination of the two. It's not mechanistically realistic as a PBPK model, but it essentially lumps the components of the system into entities that behave together similarly. And so this could be based, like I said, in time, location, or a combination of the two. And so we don't necessarily talk about space in this setting, but we implicitly talk about spatial distribution via changes that occur in time. And so these lumped parameter models essentially discretize the system through boiling it down into as small as possible number of components that is not as large as PPK and is not as detailed as spatially realistic models, but it is tractable. And so these models are called, in the most general setting, compartmental models, and they can be based on linear or nonlinear differential equations. And an incarnation of compartmental models is the non-compartmental approach that is actually based on algebraic equations. So to make it a bit more intuitive, on the left, we have the PBPK model that we described a few minutes ago. And on the right, we have a compartmental models where the system has been reduced or lumped to a relatively small set of components that interact with each other, but that are now losing the direct connection with the organ system or with the actual system that is being represented. That connection is now indirect and based on uniformity of behavior and lumping of tissues together because they behave similarly, as opposed to having every tissue and organ represented in a way that is realistic and based on its behavior and mechanistic knowledge that we have. That being said, the models on the right are quite useful because they are small, they are tractable, they are fast to solve, and so they've been essentially the workhorse for pharmacokinetics for many years before PBPK models came along. Now, how do we build these models on the right? How do we formalize what this lumping process means, and how do these compartments behave when they are by themselves or when they are connected? So there are two basic hypotheses, or shall I say, assumptions that compartmental models are based on. One is that every compartment is kinetically homogeneous. So drug molecules in a given compartment behave uniformly. They have the same probability of leaving the compartment going in any direction outside of the system. And so this is uniformity of behavior. So drug molecules in a certain compartment behave in a way that is similar. Also, compartments are supposed to be well mixed. So if there are two samples into different locations in the compartment, the concentrations that we get out of those samples has to be the same. So this is uniformity of information. Every compartment is kinetically homogeneous and well mixed, meaning that the molecules in the compartment behave similarly, and also the information in the compartment is distributed uniformly. And so that's the idea of lumping. We lump together spaces that may be physically distinct, but where the drug molecule behaves in a way that is similar. And so we use that to build models that are smaller than PBPK models or than spatially realistic models. But like I said, they're much more practical. Here's another framework that we find useful in the next few minutes. When we learn about the biological system, we can split it into two parts. This is based on the idea that only some components of an intact living biological system are amenable to measurements. So there are some compartments that are accessible. They are available to the experimentalist for test input and or measurements, and these are, we call them accessible compartments or accessible pools. There are parts of the system where we cannot either insert a drug molecule or measure from, and these are inaccessible compartments or pools. This is the whole rest of the system, which is not available for test input and or for measurement. What do I mean by that? Here's a compartmental model on the left, and I highlighted the accessible pool in yellow. So that's basically the only space where we can make input, or we can administer a drug, or we can measure a drug. Everything else is invisible. And so the idea is that to develop these models properly, a structure needs to be postulated for these models based on the limited information that is available from the accessible compartment or the accessible pool. Similarly to how we would like, for example, to estimate or to quantify how many rooms there are in a house based on how many people at a party are standing in the backyard or are visible from the windows and so on. We're trying to reconstruct inaccessible features of the system from things that we can see. And the effect of biomedical system investigation is that we don't have access to all the information that we would like. And so we have to infer what happens in an intact biological system based on limited measurements. Limited measurements, they are not just limited in time or in terms of sampling schedule or how many blood samples we can take, but they're limited in a sense of where we can take them from. For example, blood plasma is the only space that we can access easily in a biomedical system context. So what is a compartmental model in this setting? Now, to sort of summarize, we started with spatially realistic models, very intractable partial differential equations and so on. And then we went to PBPK, which is a bit of a hybrid approach where we arrange organs based on how the organ behaves and what we know about the organ and mechanistically speaking, and we arrange them in a way that is realistic. And now we went down to a further simplification, which is a compartmental model, which is a collection of compartments or pools that are each one of them taken separately, kinetically homogeneous and instantaneously well mixed. So uniformity of behavior and uniformity of information. And then a arrangement and connection of a finite number of compartments is a compartmental model. And this will be subject to a specified input and a measurement location, just like the example that I showed earlier, where there was only one compartment that was amenable to measurement among the 5 that comprise the entire model. So to summarize the process one last time, we went from a conceptualization of time and space changes of drug molecules in an intact system, and we boiled it down to temporal changes only based on a certain arrangement of units in a model that we call compartments. And so this has been a very popular approach in pharmacokinetics and had enabled the discipline to quantify several aspects of drug discovery and development, to measure PK parameters of interest, to properly characterize drugs and their behavior in intact systems over the years, over the decades, really. So for those of you that are not necessarily mathematically inclined, I would like to at least make an attempt to demystify differential equations. Whenever you see differential equations, your eyes don't have to glaze over. This is just an approach to modeling rates of change, and rate of change is a fancy name for slope. And derivative is a fancy name for slope. So we write these differential equations to model or to represent how a certain time profile changes. And so these models are based on differential equations because drug concentrations in a system, for example, change with time. And so these tools are perfectly amenable to represent temporal changes that occur in the systems that we've been talking about. And so just remember, the differential equations or derivative is a fancy name for a way to describe rates of change or essentially slopes. And we'll have more to say about that in a few minutes. I think it sort of helps to walk through a very simple example of how we build these models. And so, like I think I've alluded to, these models have 2 components mainly. One is the underlying model of the system. The system exists independently of how we experiment on it. So the system exists independently of where or whether we introduce a molecule in the system or where we sample from and so on. That's basically a summary of the principal components of the biological system. And then on this model, we superimpose an experimental design, a certain input and a certain measurement function. So for a drug, the most common example is, for example, an oral or intravenous dose of drug, either by mouth or into the vein. And then the measurement will be a cannula that essentially samples blood samples over time from a certain location, say in the forearm, right? So, but that system exists and moves in time, changes in time, independently of the experimental design that we have in terms of input and measurement. And so we show this in practice now by building a very simple compartmental model. And in fact, this is the simplest compartmental model, which is where the distribution of a drug in the body is summarized by a single compartment. That is a single space where drug molecules obey the principle of uniformity of behavior, uniformity of information. And so the idea here, this is a good model for the drug, and some drugs actually are quite well described by a simple model such as this, where the body is essentially a single compartment. And so when we write this differential equation here on the left, what I'm saying there in mathematical terms is that the rate of change of the amount Q1 in compartment 1 is equal to whatever enters the compartment minus whatever exits the compartment. So in this case, the compartment has a clearance or it has an elimination rate, which we call K01. And then that times the amount in the compartment describes how quickly drug will leave that space once it is administered. And here's my administration. It's a dose in the compartment. Now I have a new component in my differential equation, dose of T. Dose of T can be a pulse, can be a bolus, an infusion, or a more complex pattern of administration. But the amount of drug in the compartment, the rate of change of that amount, is given by the balance of what goes in the dose and what goes out, which is essentially this elimination process that is described by this rate constant K01 times the amount itself. And the last ingredient of the model is the measurement, often shown as a dotted line from the measurement location. And usually when we work with drugs, with pharmacokinetics, we measure concentrations. And so the concentration in this case is the amount Q1 divided by a certain volume, which is the volume of distribution of compartment 1. And so I just throw it in here. These entities that I'm talking about, K01 and V, these are essentially parameters, and they describe and they shape, the behavior of Q1 of T depending on their values, right, and depending on the dose and the input. For a simple system such as this one, it has a simple solution. The solution of a single compartmental model is a single exponential time course. So the time course of drugs described by compartmental models obeys exponential law. And so the concentration of drug in the compartment will decrease exponentially, and I have it there on the right. And these parameters will be estimated, K01 and V, will be estimated by matching the mathematical prediction of the compartment, which is the line, to measure data, which are the squares in the graph that you have in front of you. And so we can use matching the model to data to derive parameters from the model equation. So the best fit of the model to the curve will provide values for the parameters K01 and V. And then I can use K01 and V, for example, to calculate things that interest to me, like, for example, clearance rate. Clearance rate of a certain drug is V times the elimination rate, and has units of volume per unit time. Units are very important in this business, as you can imagine. Whatever units of measurement you use in your equations, which can be much more complex than this, need to be consistent. And so while V is intuitive, is basically, it can be, for example, liters or milliliters. K01 is inverse time because it's the rate. And clearance, which is a product of K01 and V, is liter per hour or volume per unit time. So the volume cleared in the unit time of the drug. And so you can see how helpful, these approaches can be, because first I had, 4 data points, measurements over time of drug behavior, and now I have numbers that I can use not only to describe that curve, but also to compare it to others. For example, if I have several different drug candidates, I can compare their clearance rates, their volume, and based on criteria that I have independent of that, I can decide which candidate may be better than the other to develop, and so on. I'll have more examples of that later. And so, like I said, the solution to a compartmental model is an exponential curve. And this exponential curve can be described by parameters. And in this very simple example that I walked you through, the parameters are clearance and volume. And the curve is determined by two parameters. So I can choose to report, for example, clearance and volume, or K01 and volume, or K01 and clearance. I can choose any combination of any pair of these parameters that I think is most useful. And so some people talk about primary versus derived parameters. So people would sometimes report the model in terms of clearance and volume because they're more intuitive in terms of volume clear per unit time, and volume itself as a very intuitive interpretation and mechanistic interpretation. K01 is usually a little bit harder to grasp, this sort of rate of decrease. So we tend to think of it as a derived parameter. Another useful parameter is, for example, half-life, which you, I'm sure, have heard in different contexts, is the time it takes for the amount to decrease by half. And so this is a very useful parameter that we can derive from these models to help us not only determine how quickly the drug leaves the system, but also, for example, what the dosing regimen should be, how often should we dose the drug, once a day, twice a day, 3 times a day, once a week, and also to compare drugs one to the other in terms of half-life. So now, this is a very simple model, right? How can we make it a little bit more complex, right? Some of these kinetics can be nonlinear, meaning nonlinear in the sense that the elimination rate, in this case K01, is not necessarily constant, but for example, depends on concentration. Like, for example, what if K01 has a Michaelis-Menten saturable shape as opposed to being a constant? So this gives rise to interesting behaviors, right? So, and some of you may have seen this before, certain drugs are cleared using, according to such a model. So these models, nonlinear models, end up being essentially the combination of several different behaviors all in one model. Like, for example, when concentration is much lower than the Km Michaelis-Menten parameter, then the compartmental amount will decline at the rate proportional to it, which is first order kinetics at low concentration. And then when Km is, when concentration is much larger than Km, then you will have something called zero order kinetics, right? But this is the same model. It just behaves differently depending where you are in the concentration time curve. So we can have an example. I can show you what this means in terms of how this essentially looks like in practice. So if I were to solve this model and simulate it for certain instances of the parameter values, I would see exactly what I mentioned earlier. That is, for large concentration values, which are larger than Km, the kinetics is zero order. So it basically declines at a constant rate independent of concentration. And then when I approach the Km and fall below it, then the time course becomes, the clearance becomes first order. So it's linear on the logarithmic y-axis. So you can have several kinds of behavior, several levels of complexity. With nonlinear models, you have all this in one place. So the model has a range of behaviors, a range of complexities, depending on where you are in the concentration time curve. And this is a very flexible tool. These kind of models can be very flexible to accommodate the very broad range of behaviors that we can see in biological systems in practice. Now, these examples are based on a single compartment, but of course, we can have models with several compartments where we arrange these homogeneous pools together, and with exchanges between them. They can be single directional, bidirectional, and so on. And so we connect them using these fluxes. And so these are essentially movements. These fluxes can be either physical movements among the various compartments, or it could be a composite of metabolic activity, biochemical transformation, transport. The important thing is that they occur within a similar time frame, so that the compartments, when they're arranged, they are defined in a time frame that is common among them. And again, for the mathematically inclined, this can be written in terms of differential equations, where in the end is relatively simple. It all boils down to accounting. You have the rate of change of the amount in a given compartment, which is everything that goes out plus everything that goes in. And so these equations can be written for these models in a relatively straightforward form, and people have been doing this for a long time. Luckily, these days, there is software that does it for you, so you don't have to be concerned with the details of these differential equations or writing the arrangement yourself. There are several ways to do that, which are very simple. And if we simplify the system further in a way that, say, all the transfer rates are constant, then these boil down to ordinary differential equations in steady state. And if the assumptions that I described earlier in terms of good mixing and kinetic homogeneity apply, then the differential equations can be further simplified, where the Kij in front of the Q's are all constant, and then the equations are quite straightforward to solve and write, and essentially they boil down to a system of ordinary differential equations that can be written in matrix form. And just so that you, this is an interesting feature of this class of models. If I write the Kij, the transfer rates, in matrix form, and I take the inverse of that matrix, I basically have a description of the residence time of a drug molecule in every compartment in the system. And so this can be useful if you're interested, for example, to estimate how much time a drug molecule would spend in any one of those 1, 2, 3, 4, 5, n compartments that you have defined. And this was a quick overview of this technology. And of course, the field has been moving very quickly, especially over the last few decades. And these days, compartmental models can be very complex and very detailed. Like, for example, for biologics, for monoclonal antibodies, you not only have to deal with their kinetics in the center, which often obeys a two-compartment model, but also immune complex formation and disposition due to anti-drug antibodies, and disposition based on target-mediated drug disposition, which is another way the monoclonal antibodies can be cleared. And so this class of models has a lot of flexibility and can accommodate really detailed complexity and biological plausibility, all the way without going to the mechanistic detail that PBPK models have, or to the rigorous detail that spatially distributed models have. Like I said, it's a bit of a compromise. So compartmental models are in their essence a postulation of how one believes a system actually functions and behaves. But the model is not only limited to the compartment structure and their arrangement. It also requires declaring, right, what the experiment to be performed on that model is. Like I have here, for example, a 5-compartment model, 5 pools that are distributed in a certain arrangement, from left to right, stomach, gut, plasma, and extravascular pool. So I can, for example, explore this system using an intravenous dose directly in the plasma compartment. This will bypass entirely the stomach and gut model. So whatever data I have from this experiment will not be informative on whatever happens in stomach and gut because they're being bypassed. On the other hand, I could choose to experiment on the system using an oral dose. And so in this case, my oral dose will be given in the stomach, will traverse the gut and appear in plasma. And the sampling, the measurement is still done in plasma, and perhaps even in one more extravascular pool. The time course that is derived from this particular arrangement of experimental design is very different than what can be derived from these other experimental arrangements. The location of the input is different, and they will give rise to different behaviors, but the underlying structure of the system is unchanged. The actual biological system that is being studied doesn't change. What changes is just the arrangement of input and output of dosing and measurements that I choose to probe it. And so one word about experiments. So the idea of experimental design is that I have this conceptual model that represents the biological system. I want to experiment on it. And that conceptual or computer experiment has to be the same or similar to the one that I use to generate the actual data. So in terms of input and measurement. And like I mentioned earlier, it's crucial here that we account for units properly. So for example, if dose is in mgs, in milligrams, concentration will often be in milligrams per milliliter. And so every one of these models and model parameters have units that describe how they behave and how they change. And so this accounting for units is crucial for building these models correctly, and especially to interpret their output correctly. And so there's a whole discipline built around parameter estimation. And I did mention this a little bit earlier. So in terms of matching the model prediction to data, conceptually, there are infinite values that I can choose for K01, V, and clearance, and so on. But only a relatively small set of these values will describe my data appropriately. And so that's basically how we estimate models. Right? We estimate model parameters. We calculate the output of the mathematical model. We match it to data using parameter estimation. And for the statistically minded, it's a form of nonlinear regression most often. And we extract values that then we can use, like I mentioned earlier, to summarize the complex behavior, to compare drugs to one to the other, to evaluate, for example, different formulations or different modes of administration and so on. And so the technical details are probably for other lectures in this course, but there are techniques of model building and model selection and estimation methods that one can spend a lot of time on, but we won't today. We're just interested in the output or the result of these technologies, which are the model parameters. And like for the purpose of this lecture, these are pharmacokinetic parameters: volumes, clearance, residence times. Remember what the body does to the drug. And I think I mentioned very briefly how we can rearrange or reparameterize the model parameters, for example, from rate constant to clearances, to make them more suitable to biological interpretation. And so this brings me to non-compartmental analysis. So maybe some of you are wondering, well, it's a lot of work, it's a lot of assumptions, and it's a lot of calculations that we have to make to estimate some of these parameters. Is there a way that I can do it without having to postulate these complex structures, right? So this brings me to non-compartmental analysis. So the question here is a compartmental model always required to estimate selected PK parameters from a certain curve? And so then the question is, probably not. So which PK parameters can we estimate based on only the measurements in the accessible pool, the accessible portion of the system that is amenable to measurement? And which parameters are, can we estimate with a modicum, with a minimum number of assumptions, and what these assumptions are, right? I mean, clearly, every time that we estimate something that cannot be directly measured, a model is required. But in this case, a model is just a conceptualization of how the system works. And so non-compartmental approaches have assumptions that limit their applicability in some way, but we'll show in the next few minutes how they relate to the compartmental models that we have described. So main message here is that between compartmental and non-compartmental analysis, the only difference between the two methods is how they choose to describe the non-accessible portion of the system. And you may remember, when we built a compartmental model, we have to make assumptions on the portion of the system that we don't see. We have to describe the compartmental arrangement, how many compartments are there, how are they connected, how do they clear to the environment, and so on. And we have to do it only based on measurements in the accessible pool, in the accessible portion, right? A non-compartmental model doesn't make any statement on that. It basically says, it basically focuses on the accessible pool, right? And makes all its inferences on PK parameters based on those measurements. And this is a very general statement of a non-compartmental model that can be used for like endogenous substances, for example, like hormones or metabolic substances. But for drugs and xenobiotics, the only input is from the outside. There is no endogenous production of drug, for example. Most of the time it's a new chemical, so it's not produced. And so non-compartmental model is essentially boils down to an accessible pool a system which we don't make any statement on, an elimination rate, and an input and a measure. So the accessible pool of a non-compartmental system essentially has characteristics that are the same as the compartments that we have described. So the accessible pool has to be kinetically homogeneous and instantaneously well mixed. And most of the time, I mean, it's pretty obvious by now, the accessible pool is plasma, is the circulating blood, which is an ideal compartment because most of the time it can be described very well by being kinetically homogeneous and instantaneously well mixed. And so the non-compartmental framework partitions the system into an accessible pool where we dose and we measure from, and a system that we make no statement on, right? Anything else is undeclared, right? It's something that we don't necessarily model explicitly, right? So here's a comparison of the two modeling methods, and I've highlighted the accessible pool in both. So for the compartmental model, like I mentioned earlier, just to reiterate, everything that talks with or communicates with the accessible pool is explicitly stated. In this model, there's 5 compartments. Some of them communicate bidirectionally with the accessible pool, some of them unidirectionally, and some of them clear to the environment, and some others don't. The non-compartmental system essentially summarizes the whole system into an accessible pool and the rest of the system, which is left, like I said, undescribed and undeclared. And so, but there is actually one assumption that the non-compartmental framework makes on the rest of the system. And this assumption is that there can be no additional sources or sinks within the system except where I make my measurements or where I administer my dose. So this whole other part of the system that communicates with the accessible pool cannot have any extra elimination, extravascular, for example, elimination, or any additional sources that are not accounted for in the accessible pool. So essentially, this recirculation arrow here means that anything that leaves the accessible pool, except through that irreversible loss that we see in the graph, has to come back. So there cannot be additional sinks, so to speak, within the system, except where measurements are made. And so, quite simple, right? It's a relatively simple model with respect to having to declare the individual components of the system that we have here, like in the compartmental framework. So if we use this framework, what can we estimate? It turns out we can estimate quite a bit if the assumption that I mentioned of no additional sink and source are met. So we can still estimate clearance rates, we can estimate volume of distribution and new residence time. And the way that this is done is that these parameters are estimated directly from the measurement without necessarily having to fit a model or without necessarily having to make assumptions on the unseen part of the system using algebraic techniques. And so if there's no additional sink, additional source, if the kinetic parameters are constant, then I can estimate them using this technology. So for example, what about clearance rate? The formal definition of clearance rate, as I mentioned earlier, is the elimination rate divided by concentration, but it can be shown that if the non-compartmental assumptions are met, I can have a pretty good estimate of clearance by doing dose divided by area under the concentration curve, AUC. And so this is a relatively simple calculation that can be made just based on the measurement that you have there on the right. And if these assumptions are met, can give you a pretty robust and useful estimate of clearance rate without having necessarily to declare the interconnections between the accessible and inaccessible portion of the system that we have to make in compartmental models. I will mention, though, I will start mentioning here, and I will mention it again, that when we use this relatively simple framework, of course, we lose something. If we don't go through the effort of developing a more mechanistic model, then of course we don't have the tools, for example, to extrapolate beyond the time, the range, the time range of the experiment. We don't have tools, for example, to scale between species and so on. But for an estimation that can be useful in terms of decision making and preliminary characterization, this is actually a pretty good approach. And so several calculations can be made based on this area under the curve that we talked about. There's actually two areas under the curve. One is the relatively straightforward area that is delimited by the trapezoidal interpolation of the concentration curve. The other one is something a bit more esoteric, which is the area under the moment curve, which is the area under the concentration times time. And that's used actually to calculate another non-compartmental parameter, which is quite useful, which is the mean residence time, MRT. And we can think of MRT, or the mean residence time, as the average time that a molecular drug spends in the system. Intuitively speaking, it's the center of mass of the concentration time curve. And so this gives you essentially a typical time that a molecular drug will be in the biological system post-administration. And this calculation that you have here on the screen may seem a little difficult, but let me tell you that modern software tools do it almost instantaneously. So that's another very useful parameter that I can estimate. And in this case, it doesn't require me to make assumptions on, like I said, the part of the system that I'm not measuring from. So what is needed for these estimates? Essentially estimates of these area under the curve and under the moment curve. And so these are quite straightforward to obtain with modern software. Just to sort of tell you a little bit where these numbers come from, because you will be using probably these softwares if you work in this area. So it's helpful to understand a little bit where this comes from and what the underpinnings are in terms of these estimations. Integral is a fancy name for area, all right? Just like derivative is a fancy name for slope, integral is a fancy name for area. And so these areas that I talked about can be estimated using integrals, and typically these would go from 0 to infinity, to infinite time. Of course, no experiment is conducted for an infinite time. And so we have these areas use a mix of interpolation and extrapolation. Interpolation to calculate the area that is given by the data that I measure, and extrapolation to calculate the area that is left, all the way to infinite time. And so people sometimes refer to non-compartmental analysis as model independent. That is true in a way, because one doesn't have to make a lot of assumptions on the inaccessible portion of the system. So model development is at the minimum, but there is at least a modeling assumption or statement that is made when we extrapolate beyond the confines of the experiment. So when we go to infinite time, you need a little bit of a model to do that. And like we'll see in a minute, the most common of that is essentially a single exponential function that is extrapolated all the way to infinite time. And so these, like I said, for interpolation, when it comes to calculating the area under the curve that I measured, we can use various methods, and these go by the fancy name of trapezoidal, log trapezoidal combination. The trapezoidal rule is nothing but an estimate of the area based on the, quite literally, the area under each pair of data points that is connected by a straight line. And then you calculate the area under that particular trapezoid, and then you sum them all up, and you have the area under the curve up to the last time point. You can, for curves that change, maybe they have a different pattern of change, for example, rising and falling. Log trapezoidal is a little bit more useful, but this is just math, and I think that suffice to say that there are ways to estimate the curve under the data that we measure, right? So these are, it can be as simple as calculating the area under each trapezoid and then summing them up. And then, of course, I need a way to extrapolate further, right? So extrapolation is always necessary. But let me just say, before we go to that, if I estimate the area under the curve with very limited data, like the one on the right, chances are my estimate is not going to be very precise. Like in this case, for example, if I sample only at 1 hour and say 9 or 10 hours, then I'm missing all the behavior that is in between. And in this case, I'm going to overestimate my area under the curve because I'm actually getting a lot more area than the actual data would have given me had I been able to sample it. So as the number of samples of blood sample goes down from dense to sparse sampling schedule, then interpolation, even interpolation may not be accurate. It depends on the shape of the curve, and there are considerations of experimental design that help us make an educated guess as to how to optimally place these samples, right? And that's another field that has been evolving quite a bit in the last few years. And like I mentioned earlier, because these areas are needed for infinite time for the formulas to work, once we take the last measurement, we need a way to extrapolate beyond the end of the experiment. And so most often, the terminal portion of the curve where I haven't measured is assumed to be a non-exponential function, a single exponential starting from the last point onwards. And so it has an exponent and a rate of decay. And of course, I can also estimate from there the terminal half-life, the half-life of terminal decay, which is another important component of non-compartmental analysis, and is often reported in these kinds of analysis. So the half-life of terminal concentration is as simple as log 2 divided by the exponent of terminal decay, which can be calculated from data without having a more complex model than just a single exponential going down with time. And so, like in this case, from the last data point, I can essentially extrapolate with a single exponential. Here is that portion of the curve that starts at the last yellow diamond, and that's basically all things that I haven't measured, but I can extrapolate, and that can give rise to an estimate of half-life, which is essentially the terminal half-life of the particular curve. Now, this extrapolating function is crucial. If, for example, I didn't do a good job in experimental design, maybe I stopped sampling too early, and now I have a little bit of an ambiguity. Which curve is the right one? Is it the green or the red, right? They both fit data properly up to the last point, but the long-term, the estimation of the area under the curve that is under the green line as opposed to the red line, they're quite different. And so the choice of extrapolating functions is crucial to get an accurate or realistic estimate of area under the curve. And there are techniques that we use in terms of including an optimal number of data points along the curve to choose what function may be better. But of course, it is a source of potential error, because if I leave the extrapolation too ambiguous, then my estimate can be wrong. And just to remind you, why do we use, for example, the area under the curve? Is to provide us with an estimate of clearance. So if my AUC is wrong, my clearance is going to be wrong. And so I may make the wrong conclusion in terms of how the drug behaves in a biological system. And so, like I said earlier very briefly, when we estimate the required integrals, we just sum up the individual components. So we sum up the part of the integral that is from trapezoidal integration, and then the last element in the sum is the extrapolated time concentration curve, which then completes my area under the curve and area under the moment curve calculation. And then I can use these now to calculate my non-compartmental parameters, including, like, for example, clearance, mean residence time, and so on. And then half-life was also a byproduct of this. From the extrapolation, I can get an estimate of half-life. And the beauty of this calculation, is that extrapolation is done automatically as part of the fitting, and one can have statistical information for all the parameters. And despite the simplicity of this approach, there is a natural connection with the solution of constant coefficient compartmental models, if you're interested. And software is widely available for those of you that don't want to deal with the intricacies of model building, but that prefer essentially to have a reproducible compartmental system or computer system calculate these for you. But despite the beauty and the elegance of the approach, there were some assumptions. And so if you remember, the key assumption that we made based on non-compartmental analysis was that every molecule that leaves the accessible pool has to come back, except when it's irreversibly cleared from the accessible pool itself. So depending on the particular drug and the particular biological system, this assumption can be met or not. But if it is not met, then there are the estimates will be incorrect, to a varying degree. And also the last item that we mentioned is that if there are non-linearities in the system, like the time varying rates that we saw earlier in the compartmental case, where we had the Michaelis-Menten rate of elimination, the non-compartmental approach cannot handle those cannot handle those properly. And so that means that in this case, non-compartmental analysis cannot be applied because the estimates may be misleading. So the advantages of compartmental models in general are that they can handle nonlinearities more easily. They provide a way to state hypotheses that we may have about the system structure and the arrangement of these compartmental units, can aid in experimental design, and can support translational research. But the non-compartmental method has an advantage that is very easy to calculate and can provide very useful parameters if the assumptions are met. There are the biases that can be introduced by non-compartmental analysis have been described previously, and these essentially boil down to violation of the single sink and the single source hypothesis, and these have been described in the literature, but tough is to say that if the assumption on the right is not met, then some of these estimates may be erroneous or misleading. So one instance, for example, where non-compartmental analysis can be misleading is that of biologic or monoclonal antibody pharmacokinetics. Often, and I think you will see it in other modules of this course, but often antibodies exhibit what's called target-mediated disposition, which is basically the receptor, the target receptor itself impacting the disposition of the antibody at the target site. And essentially, the target, if it turns over quickly and is present in a large abundance, can actually turn into an additional sink for the antibody, meaning that the clearance of the antibody will be impacted, will increase because of disposition that occurs through target binding. So the elimination of the antibody drug can occur at sites that are remote from plasma due to binding and internalization processes. And so this is basically boils down to a violation of non-compartmental analysis, because when drug elimination is influenced by binding to the target, the assumption may not be met and parameter estimates may be misleading. And moreover, such a system doesn't necessarily meet linearity and time invariance. But that being said, we can still use non-compartmental analysis to figure out that something is going on. Like in this example, we have, if we plot non-compartmental AUC volume, clearance, and half-life as a function of dose, we see that the system doesn't behave linearly with respect to dose. And so this is a signal or evidence that non-compartmental analysis assumptions cannot be met, and the AUC and clearance and volume at any given dose are not a complete representation of the disposition of the drug in this case. And so one has to resort to more complex modeling approaches where the PK of the antibody includes not only administration and distribution, but also binding to the compartment R, to the pharmacological target receptor, and the resulting elimination that occurs from there. But again, non-compartmental approaches can be helpful to help us understand that something is going on, and a more sophisticated method of analysis may be required in this case. So there's a lot of literature on these ideas, and there's a lot of writings that one can go to understand this a little better. I put some of them in the lecture. But maybe if there is a take-home message here is that to estimate key useful PK parameters. Either approach, compartmental, non-compartmental, is probably adequate, when the sampling schedule is dense or rich, provided that all assumptions that are required for either analysis, non-compartmental especially, are met. If the sampling schedule is sparse and the disposition of the drug is non-linear, then these are challenges for non-compartmental analysis. And then when it comes to non-compartmental methods, maybe the last point is that they cannot be predictive. They cannot be used to scale from one system to another or from one species to another, like compartmental models can do under some circumstances, and PBPK models do very elegantly. And so they have limitations in that sense. So the best strategy, I would say, is probably a blend. So it's perfectly okay to start data analysis with the very simple non-compartmental approach and see what the numbers are, and then see what the behaviors are. If there is evidence for nonlinearity or saturation and so on, then one can switch to other approaches, always being careful about the key assumptions. So I think we got to the end of this particular module. So thank you very much for listening so far. And I'd like to acknowledge again David Foster, my former colleague, who was the one that developed these methods back when the course was actually started. So thank you very much, and good luck with the rest of the course.